Hi, everyone. Thank you so much for being here. This summer I had the opportunity to work in the Davis lab, and it's been a really gratifying experience. Today, I'm going to be talking with you about unraveling the role of GBA in Parkinson's disease progression. To begin, I want to address the widespread reach of Parkinson's disease throughout our ever-growing aging population. It's the most common neurodegenerative movement disorder disease, affecting 1% to 2% of the population over the age of 65. Typically, people think of Parkinson's as just a movement disorder, but it's more than that. It's also a non-motor disorder with cognitive symptoms such as sleep disturbances and cognitive decline. The most important neuropathological finding in Parkinson's disease is the presence of Lewy bodies. These are neuronal protein aggregates that consist of ubiquitinated proteins, alpha-synuclein, and lipid membranes. The degeneration of dopaminergic neurons throughout the brain is also something that's very crucial for Parkinson's disease. The figure down below shows something that is very important for understanding what I'm going to be talking about. The clinical progression of Parkinson's disease correlates with the stereotypic spreading of Lewy bodies throughout the brain, and the spread of this protein correlates with clinical progression. And on the right, you can see an image of a Lewy body. The risk factor GBA is known to be the strongest genetic risk factor for developing Parkinson's disease. And this is important because it increases your risk of developing Parkinson's disease by up to 5 times, and approximately 10% of Parkinson's patients have this mutation. Other mutations, such as common polymorphic variants, have little effect on developing the disease, so they're not as clinically relevant. You're probably curious what GBA is. It's a gene that encodes a liposomal enzyme, glucocerebrosidase, and this is important for lipid metabolism. And specifically, it breaks down glucosylceramide into glucose and ceramide. In addition, our lab and others have found that GBA carriers with Parkinson's disease have shown faster clinical progression over a time course of a couple of years. With this faster clinical progression of Parkinson's disease and my mentioning earlier of how the spread of proteins correlates with clinical progression, we're curious why this protein spread is happening quicker. So this is what I'm going to be focusing on. In order to understand the mechanisms of GBA mutations, we use both a Drosophila model and an iPSC model. Caroline Kwan over there, who was in this program last year, used the Drosophila model, and this summer I used the iPSC model. With these methods in mind, we were interested in how GBA mutations accelerate the propagation of this Lewy pathology and how they alter extracellular vesicle composition. First off, what are extracellular vesicles? Extracellular vesicles are small lipid layer enclosed vesicles secreted by most eukaryotic cells. They're really important for intracellular communication.
Sorry, they're not seeing the slides online.
Cool. Okay, so extracellular vesicles are secreted by all types of cells, and they're very important for intracellular communication. In addition, they're hypothesized to be the main spreader of protein aggregates throughout the brain. So in order to study extracellular vesicles, we have cell cultures of neurons from a healthy control, a Parkinson's patient that is age and sex matched with the GBA mutation, as well as an isogenic control line where this GBA mutation is repaired through the CRISPR-Cas9 system. I'm able to isolate vesicles from the liquid these cells are in, because earlier I mentioned that all cells secrete extracellular vesicles. And then I'm able to use filtration as well as size exclusion chromatography to concentrate and further remove impurities. To visualize these vesicles, we use this really cool technology called ZetaView, which is a nanoparticle tracking software analysis. This allows us to detect the size of the vesicles as well as the concentration. In this slide, you can see results that I obtained using the ZetaView to look at the size of the vesicles for our wild type and our GBA Parkinson's disease patients. We observed no statistical significant difference, but it's really cool to be able to observe the different sizes of the vesicles since they're really small biological systems. In order to characterize EV-associated proteins, we used Western blots. I didn't have a chance to do my own Western blots yet, but I'm working in the lab next year, so I'll have a chance to do that. But on the left, there's an example of a Western blot showing that we can detect ubiquitinated proteins. And it's important for us to be able to characterize what proteins are in these GBA mutant extracellular vesicles, as well as how this mutation changes lipid metabolism further. In the future, we're looking at a hypothesis that alterations in extracellular vesicle composition due to GBA might be leading to the seeding of pathogenic protein aggregates in recipient cells. In order to test this, we're going to have cultures of GBA Parkinson's neurons as well as control neurons, and we're going to be adding vesicles from mutated cell lines, and we're going to observe the uptake of these cells. Will this lead to cell death? Will it lead to endolysosomal trafficking defects, or will it also lead to the aggregation of endogenous alpha-synuclein? I'd like to say thank you to my lab, as well as my PI, Dr. Davis, for this truly amazing experience, as well as my lab members sitting over there. This has been a really great experience, and I'm so grateful for you guys welcoming me into the lab. In addition, I'd like to say thank you to the neurosurgery department, as well as Dr. and Mrs. Ellenbogen, as well as Julie and Sylvia for making this a truly memorable experience.